Justin Chitpin from the University of Ottawa. So, in this talk, I'm going to be giving, first of all, a bit of background on what metabolic flux networks are and how they've been analyzed using what I call molecular elementary flux modes, or EFMs. This is going to lead to two problems in EFM literature I'm going to cover, notably calculations associated with enumerating these EFM pathways, and something we'll talk more about, which is decomposing fluxes onto EFMs. And I'm going to show how we can solve both of these problems through our proposal of what we call atomic EFMs and our algorithm, the atomic cyclohistory Markov chain. So first of all, when I'm talking about metabolic flux networks, I'm talking about knowledge-driven networks. So we take our metabolites, represent them as nodes, and those nodes are connected to each other by directed edges corresponding to, say, the protein enzymes or maybe even transporters that move those metabolites within different subcellular compartments. So these fluxes correspond, well, the edge weights are assigned to metabolic fluxes. Those are really the biochemical reaction rates of those enzymes and typically are estimated under some form of steady state. That implies, of course, conservation in the metabolite inflows and outflows with respect to time. And there's a variety of ways these can be experimentally or computationally estimated and inferred. So in recent years, there's been a bit more metabolic flux data that's been generated, like Nielsen now, who were able to estimate a little over 500 steady-state fluxes from a combination of absolute protein and metabolite abundance data and some computational modeling. So this led us to ask the question of how we can use this type of metabolic flux data to make predictions about the fate of individual molecules or metabolites as they enter the cell and are remodeled. So that particular question has historically been tackled through this concept known as the elementary flux mode, or EFM for short. An EFM is essentially a very special type of steady-state pathway through a network. So through the series of reactions, we balance our molecular stoichiometries, hence the steady-state bit, but they're also considered non-decomposable or minimal pathways. And that simply means we can't take an EFM and represent it as two or more smaller pathways carrying steady-state flux. So to give an idea of what they look like, if we consider purely unimolecular reaction networks or metabolic graphs, we can see that an EFM could be a source to sink pathway through the network, so that kind of simple path. It could be a simple cycle within sets of internal metabolites. And of course, if we have multi-species reactions, then that idea of source to sink flow or internal simple cycle can extend to account for possibly multiple inputs, outputs, or branching reactions to maintain that cyclic flow. And so what's nice about EFMs is for any set of steady-state fluxes we observe in a network, we can always represent those as some positive linear combination of their EFMs. So we refer to this problem as the EFM flux decomposition problem, the idea being that you have a network like the one on the left, and you'd like to know how, given those steady-state fluxes, how metabolite 1 travels through to arrive at metabolite 6 and leave the network. Well, for many biological networks, we often have many more EFMs than we have fluxes or linearly independent ones. So of course, we could say maybe have an explanation with 2 units of the red and blue EFM, or 2 units of the purple and orange, or some positive linear combination of all 4. That ambiguity, of course, is problematic because these are really mechanistic pathways that should be used to predict how metabolite 1 flows through the network. If we have multiple explanations, we lose that predictivity. We can't do any form of differential, say, downstream analysis if we have multiple conditions. So to solve this problem, we turn to this idea of redefining EFMs. Now, in literature, they've been defined with respect to balancing molecular stoichiometry, so I've often written or referred to them as molecular EFMs. And so one of the challenges of molecular EFMs, the standard definition in the literature, is they can be really complex pathways. In big networks, we can have multiple inputs and outputs, as you can see by our molecular EFM highlighted in red. And so when we're interested in studying metabolism, we're often interested in studying the nutrient source remodeling metabolism. And so why not instead define an EFM with respect to, say, an individual atom, like a carbon of that glucose molecule entering the network and tracing that possible steady-state flow as we see in the atomic EFM shown in blue for a given glucose carbon. So this leads to linear pathways, which are a lot easier to interpret, even as the network scales in size. And that linearity is also quite nice because last year we published an algorithm on solving that EFM flux decomposition problem, strictly for the case of unimolecular reaction networks. So the idea was we took our metabolic network, a graph in this case, and we modeled it as a discrete time Markov chain where we set our transition probabilities proportional to the steady state fluxes of one metabolite undergoing some type of remodeling event to become another one. And by expanding our notion of state to not just be a single metabolite, but sequence of metabolites, we constructed what we call this cycle history Markov chain, which allows us to very explicitly keep track of these metabolite movements on its way to completing a simple cycle going up that tree. And each one of those simple cycles highlighted in red, orange, purple, and blue, well, they correspond to a given EFM. And so in order to extend this framework to atomic EFMs, it's actually rather simple. All we need to do, one second, is include our 3D structures of our metabolites. Those can be encoded as smile strings, and using programs like Reaction Mapper, an atom mapping tool, for any given reaction equation, we can determine where that atom moves from the substrate side to the product side of that reaction. And so if we do this for a source metabolite carbon, for example, like the one for glucose shown here, we can construct an atomic transition graph. And as a graph, we can then go ahead and construct its corresponding atomic cycle history Markov chain. That, of course, is good for enumerating our atomic EFMs. And if we supply fluxes at steady state, we can also estimate their weights under our Markov chain model. So I'll be showing some results for 5 genome scale metabolic models: core E. coli model, human red blood cell, Helicobacter pylori, Staphylococcus aureus, and that human hep G2 liver cancer cell line from Nielsen Nell that we saw earlier. And that's the only data set present today that contains a single set of steady state fluxes. And we'll be showing results purely for carbon atomic EFMs. So the first question we wanted to ask was whether it's even possible to enumerate all of these different atomic EFMs. And so using our method, Markov weighted EFMs, we enumerated between several thousand and a few million of these different carbon pathways across our different data sets. And in contrast to more standard molecular EFMs defined originally by Schuster and Hilgetag, well, as you can see, there's orders of magnitude more of them, often because of the different combinatorial ways of balancing those metabolic byproducts. Now, of course, when we look to the runtime, we can see that our method is running between 2 and 6 orders of magnitude faster. And that also includes if we take our competitor flex mode calculator, which is the state of the art for molecular EFM enumeration, and run it in parallel, where we, I think, optimize 16 to be the best number of threads for both of these data sets to have the fastest runtime. So we can enumerate these different carbon pathways. From here, I want to get a broad understanding of what type of pathways these carbon EFMs represented. So on your left, I'm showing a bar plot showing the different categories. Are they source to sink flows, explaining nutrient source remodeling? Is it an internal simple cycle? And within those two categories, stratified them further to account for cases where that carbon may revisit the same metabolite, but in a different atomic position, like doing 2 turns of TCA cycle. And as you can see, the majority of our pathways correspond to source to sink or nutrient source, explain nutrient source remodeling. When we looked at the distribution of our EFM lengths, or the number of reactions, we found that for our most highly connected hep G2 data set, that on average, the carbon EFMs went through approximately 63 reactions. And that's just interesting from a structural perspective, given how many different ways those carbons can remodel through the network, becoming maybe the same metabolite, but moving around in their positions within the different carbons. Now, focusing a bit more on the source metabolites, we wanted to ask whether the individual carbons within them could undergo different EFMs, and to quantify that. So we may have threonine, for example, entering the network. Here I've listed the total number of EFMs for each of its 4 carbons, and we're presenting the max over min ratio as our statistic here. And so for metabolites like glycine, 2 backbone carbons, they're equivalently metabolized through the same number of EFMs. But for other types of amino acids and branch chain amino acids, we can see that the number of different ways that some of those R group carbons can remodel through many more, thousands more EFMs, compared to their backbone carbons is remarkable, possibly going to become different internal metabolites, leaving the network at different spots, or even maybe going through the same sequence of metabolites, but at different atomic positions or in different orders. So moving on, beyond this type of structural analysis, we want to analyze the fluxes, or that single set of fluxes from the HepG2 data set. So we don't have multiple replicates, we don't have conditions, so we couldn't do a differential analysis. We know that HepG2 is a liver cancer cell line, like many other cancers, is addicted or glutamine dependent. So it's an interesting question to study how glutamine is metabolized and taken up into those cells. So we have many thousands of these carbon EFMs, and what we did is, on the left graphed the cumulative explained glutamine flow as a function of the individual carbon EFMs of glutamine, from highest weighted according to our model to smallest. And as you can see, the top 5 EFMs for collectively the 5 glutamine carbons explain about 50% of that total glutamine carbon flow, pointing to some really active or dominant EFMs explaining that metabolism. And so if we take, I think you can see the pointer, right? Nope. Never mind. If you see yellow, orange, the backbone carbons, here I've taken for either of them, because they have the exact same top 5 greatest EFMs, I've plotted them as a Sankey diagram on the left, where you can see that our EFMs recover some really well-known and established metabolic pathways. EFMs 1, 3, and 5 recovering the malate aspartate shuttle. EFM4s we can possibly recall from metabolism classes is the standard citric acid cycle. But interestingly, EFM2, which is the second biggest one, predicts what is now referred to as non-canonical TCA cycles, something discovered quite recently and validated with stable isotope tracing analysis, a truncated version of the TCA cycle that possibly could be an intermediate metabolic transition between high glycolysis and the more standard OXPHOS state the most healthy cells undergo. So to wrap up, currently, and to the best of my knowledge, there's not too many bioinformatic tools out there to analyze this type of metabolic flux data, at least large-scale metabolic flux data. And through atomic EFMs, I've shown today how we can not only compute these pathways on some smaller genome-scale networks, and actually solve that EFM flux decomposition problem to figure out the amount of flow through these different pathways that we enumerate. And so when we applied this method to our HepG2 data set from Nielsen and al., we saw that our EFMs make sense according to experimental studies shown in literature, but we're also predicting new metabolic pathways that hopefully can be validated for this particular cell line. So with that, I'd like to thank you for your attention, thank the GLBIO organizers, acknowledge my sources of funding, my lab, and give a shout out, of course, to my advisor, Ted, who's here today. Thank you.